This is going to be our overview of the laboratory for muscle. And this first slide shows an image of skeletal muscle. Note the very characteristic natures of skeletal muscle, the peripheral nuclei and the cross striations. Now this band of connective tissue running down the middle of the field would be a profile of the perimysium. This is connective tissue surrounding groups or fascicles, groups of muscle cells. The very pale lines that you see here would be endomysium. That would be connective tissue surrounding an individual muscle cell. We will talk later in the course about epimysium, perimysium, and endomysium. The peripheral nuclei in skeletal muscle are very evident, and I'd just like to make a point that as you look at skeletal muscle cells this way, most of these nuclei that you see would indeed be nuclei of the skeletal muscle cells. Some of them could be nuclei of fibroblasts that would be making up the endomysium, the connective tissue surrounding the individual cells, and some of these could also be nuclei of satellite cells. Satellite cells serve as stem cells for skeletal muscle, and we'll talk about satellite cells and their significance later in the course. Note within this connective tissue you can see some small blood vessels. Another view of skeletal muscle both in longitudinal and in cross section. You can clearly see the individual muscle cells, the peripheral nuclei. I think you can get a sense of the endomysium, and you can see the myofibrils, and they're especially evident in the cross-sectional profiles. And of course the myofibrils consist of actin and myosin microfilaments, and we'll talk about that later in the course. Another profile of skeletal muscle fibers both in longitudinal and in cross-section, and I think you can clearly see the peripheral nuclei in these cells, shown like this, shown here in the longitudinal section, and you can get a sense of the endomysium around the individual muscle cells. Another view of skeletal muscle both in longitudinal and cross-section. Clearly you can see the individual cells, you can see the peripheral nuclei, and in some of the cross-sectional profiles you can get a sense of the red and white muscle fibers. We'll talk about the significance of red and white fibers later in the course. This is an interesting view of skeletal muscle. It's artifactually separated. It makes the point that you can see these light areas would be areas of the perimysium that would surround fascicles or bundles of muscle cells. Around the individual muscle cells or fibers you can see the endomysium, and of course the epimysium would be the connective tissue surrounding the entire anatomically named muscle. This is just a higher power view of the slide I just showed you, so you can see the endomysium like so, and the perimysium would be like so. This is a profile of smooth muscle, and on this image you can identify individual spindle-shaped smooth muscle cells with their centrally located nuclei. In the upper left of the image you can note a small blood vessel, probably a venule. You can clearly see red and white blood cells in the lumen, and you can see the endothelial cells that line this blood vessel, and there's connective tissue around the vessel and around this portion of the smooth muscle. A mass of smooth muscle cells shown in this image, and I show this because I'd like you to keep in your mind what smooth muscle looks like in relation to dense irregular or dense regular connective tissue. You can notice in the smooth muscle individual cells that are eosinophilic and staining, and within these eosinophilic staining cells you can actually see the nuclei of the smooth muscle cells. This is an appearance that's very much different from what you'd see if you were just seeing fibroblast nuclei within connective tissue. And the point that I like to make is that when you're looking at smooth muscle, the smooth muscle is much more eosinophilic in staining as compared to connective tissue. Here is a view of smooth muscle. This section is taken through the muscularis externa in the wall of the small intestine. You can note smooth muscle fibers both in longitudinal section and in cross section. You can see the central nuclei. In some of the longitudinal sections, you can see the corkscrew or squiggly nature of the smooth muscle cells. We'll talk later in the course about why histologically smooth muscle cells take on that appearance. In the cross section, I'd like you to keep in mind the size of the smooth muscle cell and the size of the nucleus in relation to that smooth muscle cell. We'll come back and talk about that in just a few minutes. Here at the very top of the field are profiles of mesothelium, because this section is actually on the serosal surface of the small intestine. I, of course, would not expect you to be able to know that at this point in time. Here is a view of cardiac muscle. You can see the typical branched nature of the cardiac muscle cells. Clearly you can see the striations in the cardiac muscle cells. The nuclei are centrally located, and in several places on this image you can get a sense of the intercalated discs. Another view of cardiac muscle, you can clearly see the striations, you can see the nice intercalated discs, and you can see the centrally located nuclei. Yet again another view of cardiac muscle, both in longitudinal section on the bottom and in cross section towards the top of the image. Look at the size of the cardiac muscle cell as I'm tracing it like this. So the size of the cardiac muscle cell is quite a bit bigger than the size of smooth muscle cells. These are about the same magnification as the images I showed you on smooth muscle. Look at the size of the nucleus in the cardiac muscle cell, and remember that in relation to the size of the nucleus in the smooth muscle cell. And I'll make another point about that in just a couple of minutes. Here, a higher power view of cardiac muscle cells in longitudinal section. You can clearly see the striations, nice views of the intercalated discs, and this slide also makes the point. The central nuclei and the nuclei have a relatively clear area around them. This is called the perinuclear area. This area is rich in organelles, especially mitochondria, and has substantial deposits of glycogen. Both of these are necessary for the massive amounts of energy that cardiac muscle cells have to generate for their metabolic activity. A nice high power view of cardiac muscle cells in cross section. Clearly you can see the centrally located nuclei, and the stippling represents sections of the myofibrils, of course the actin and myosin microfilaments that are necessary for the contraction of cardiac muscle cells. Here would be an example of a smooth muscle cell cut longitudinally and in cross section. Here is a cardiac muscle cell cut longitudinally and in cross section. And I put this in because I want to make a point. Even though when you look at these cells, the cardiac muscle cell would be a larger cell in relation to the smooth muscle cell, what I want you to look at is the nuclear to cytoplasmic ratio. Notice that in the smooth muscle cell, the nuclear to cytoplasmic ratio is much higher than it is in the cardiac muscle cell. And so that is sometimes a good way to help you diagnose the difference between smooth muscle cells and cardiac muscle cells. Simply look at the nuclear to cytoplasmic ratio. I've shown you these two slides before, but now I put them on the same image. These are at the same magnification. Here's the cardiac muscle cell. Look at the size of the cell. Look at the relatively small nucleus in relation to the size of the cardiac muscle cell. On the other hand, look at the smooth muscle cells. Relatively same magnification. Look how much smaller the individual smooth muscle cell is, but look at how much larger the nucleus is. So the smooth muscle cell has a much larger nuclear to cytoplasmic ratio than the cardiac muscle cell. Here in skeletal muscle, a muscle spindle, so-called intrafusial muscle fibers, specialized for sensation, but these cells are much smaller than the typical extrafusial or ordinary skeletal muscle fibers. And we'll talk about these later in the course. This is a junction of a nerve with a skeletal muscle cell. This preparation happens to be stained with a heavy metal. It's gold chloride. You can clearly identify the skeletal muscle fibers, and you can identify these alpha motor nerve fibers that are terminating in a motor end plate. You can clearly see the striations in the skeletal muscle. The next image is a higher magnification of this motor end plate, and you can actually even see some of the synaptic boutons or terminal boutons at the end of this motor end plate. Here just is a myotendal junction. So here's the skeletal muscle, and the connective tissue around the muscle is blending in with the connective tissue of the tendon.